Hello, everyone. My name is Patrick Moran. I'm the founder and CEO of Pebble Life Science. We're talking to you today about a new pillar of hope in cancer treatment. Our key partners with this are MD Anderson Cancer Center, Kemp Proteins, that's a main supplier for NCI, and we started our discussions with NCI on a non-dilutive tech transfer collaboration earlier this year. The key team that has put together this seed stage biopharma is 10 years together at our core. Our scientific advisors at this point are all MD Anderson affiliated, and we have deep experience through our outside counsel in life science, patent, business development, and FDA regulatory. This is where the rubber starts to hit the road. Current pillars of cancer treatment all have dose-limiting quality of life toxicities. Most of the mainstays and combination therapies do not increase overall survival. The pillar that we've been working to establish offers monotherapy for multiple treatment-resistant solid tumor and hematological cancers. We're doing this through a dry fill capsule. It produces an ease of compliance, manufacturing, distribution, administration, and potential collaboration with the other pillars. If you look at the white text underneath the pre-existing pillars, these are based on both internal and published external studies. It talks about how we can address collaboratively some of the current standard of care limitations that are plaguing the current standard of the pre-existing pillars. That starts to highlight our key distinctions. It's all based on simplicity. There's an ease of working with it, and that's all based off of a foundation of safety for the patient before efficacy. We've done this in really quite a simple, elegant, and natural way. We took one plant and one mushroom. Both have been consumed by humans for millennia. Both have been proven to kill some cancers. So what we did is we isolated the most beneficial API out of those. We started combining those into unique formulations and testing them for the most effective ones, testing them against the current standard of care. That led to our key innovations that we've been patenting. We have a platform of novel small molecules and proteins. Many of the single agents have already been proven with neurological benefits that counter standard of care toxicities, and our two-agent combinations exceed standard of care efficacy without any of the adverse effects. In the process of determining a lead candidate, because we come from a natural product background, we're able to use high dose extracts in human case studies. And so one of the most significant was a 62-year-old woman with recurrent platinum-resistant ovarian cancer. Carboplatin, paclitaxel, Doxil, and ultimately Elahere failed her. During the time when she was just on our monotherapy protocol, we flattened her tumor growth in 20 days. We reversed it in the next 22 days, and our oncologist noted in her files a healthful appearance and vitality contrary to chemotherapy patients. And so what we did with that, going from that supplement level that was successful and moving that into an API that's ready for the FDA, we took two agents. We've codenamed them for the purpose here of C4 and F1. If you look at the chart, F1 was killing treatment-resistant breast cancer at 5%. C4 was killing treatment-resistant breast cancer at 8%. But when you combine them together, the synergistic effect is fully fivefold. And we were killing breast cancer at 63%. For ovarian, like our human case study, our kill on contact is over 90%. And so we're taking this to the FDA. We've teed ourselves up for we're in the kind of sweet spot for an FDA Interact meeting. The current data package that we're taking to do that with, NCI actually submitted their own NCI 60 on one of our source extracts, our fungal extract. It came back with multiple hits on ovarian cancer and breast cancer, in addition to other indications. Our safety, I want to stress, we do not work with controlled substances, so there's a clear regulatory pathway throughout. The fungal extract is actually already an approved cancer medication in both Japan and in China, is prescribed regularly, from which we've derived our API. On the efficacy side, our mechanisms of action are synergistic. There's multiple of them. We have a direct anti-cancer activity that's similar to the effects of chemotherapy and radiotherapy. We have inhibition of cancer cell growth that's similar to several targeted therapies, and we have an increased immunoresponse that's like immunotherapy. Over on the right side, but without any of the adverse effects and none of the toxicities. On the right side, we're far enough along in our proof of concept studies and our manufacturing that we're preparing for that FDA meeting. We've been GMP manufacturing, and we've been able to establish recombinant expression on our protein. Addressing some of the early stage risks, we've been out in the VC market for a few months. We're getting pretty consistent feedback across the board. It's an exciting, right? It's an exciting play. We're basically doing everything right. No major course correction, but in the current investing economic environment, we're too early stage for us to invest. That quotation is actually from a VC. No VC believes anymore that in vivo results translates to human efficacy. It was a heck of a thing to hear. So I'm putting forward some mitigating facts that are obvious and unique to our distinctive play of what we're developing. There's already demonstrated efficacy in humans. The source mushroom extract is an approved and regularly prescribed medication in both Japan and China. It has been since 1987. 14 Pebble executed human case studies have all shown efficacy with no adverse effects with supplement level extracts from which we directed our API. If for some reason this doesn't statistically hold up and our clinical trials would fail with the combination that we've put together on the indications, there are several single agent and combo options to take, both monotherapy and combination therapies. You can actually see this in the competitive market today. There's some unfolding. We're on an IND pathway. We can also pull back to our source extracts and choose a botanical drug pathway and pursue like what Japan and China have done if we needed to fall back. We're also in talks for licensing now for out licensing as natural product supplements. And we have multiple successful case studies for neurological conditions from PTSD, ASD, et cetera. Our exit plan is comprehensive and falls into significantly more detail than that's here. I'm going to focus on just where we are today. That's the FDA Interact meeting. We're preparing that now. By the FDA's timing, we should have our response in 1Q of 25. The seed round funds that we're raising now get us to our first in human trials on the other side of pre-IND. The outside of that is 3Q26. But if we stay on track and we get the FDA response that we're teeing up for, we could cut that by up to 2 quarters, which would put us at first quarter 26 for first in human. And that's where some of those VCs have expressed an interest in engaging in a Series A with us. Some of our highlights: robust patent protection. Again, we've got very well-seasoned outside counsel. We've been filing patents since 2021. We started with provisional. We filed non-provisional, national for national and international. We filed continuation in part as we've continued to protect our innovations as they unfold, as well as tune in our indications with our lead drug. For partners, what we're looking for and they're interested in is impact investors, out-licensing beyond our human oncology specific bandwidth, and we're looking to expand our pharmaceutical and academic research collaborations. That's our contact information, a little summary of the whole thing, and I'm grateful to be here. Thank you very much.
Great presentation. Thank you so much. So just to make sure, is this a mixture or just one compound?
It is a 2-agent combination drug, 1 small molecule, 1 protein.
What is the mode of action?
There are multiples. If we go back, we've got over here, synergistic multiple. So we have direct cytotoxicity. We have EFGR response, increased immune response, inhibition of cancer cell growth. So there are multiples that are coming together. It's a deep dive question. Is that what am I addressing it?
Not really, but okay.
Can you ask it in a different way, maybe?
Well, so at the molecular level, what do you see? What pathways are they affecting? Because you're addressing it in a general way. So I was just wondering what the mechanism of action is at the molecular level.
I am addressing it in a general way. So I am not the scientist on it,
okay.
But I can put you in contact with the folks at MD Anderson.
And what's the mushroom? Which one is the mushroom that you use?
We use Trametes versicolor.
Thank you so much.